and thank you everyone for attending today. Today I'll be talking about some work we've done in close collaboration with our partners at SCIEX to create an automation-enabled LCMS analysis of biologics. So biologics is a very vague term, but it typically seems to refer to things that are not chemically synthesized. So again, for folks in the LCMS space, typically we think in terms of small molecules. Biologics tend to be significantly more complex and larger molecules, and typically they're referring to recombinant proteins. And what you can see from this somewhat slightly dated information here is that there's over 1,000 biologics in development as recently as 2012, and that seems to be increasing. And back in 2012, that equated to a $60 billion market, with about 40% of that market coming from monoclonal antibodies. That's continued to grow, and it was estimated that it would be a $190 to $200 billion market by 2015. So again, just kind of reinforcing the fact that biologics are a very essential and growing portion of the biopharma market. So what does that mean for LCMS analysis? Well, again, as mentioned before, you can have molecules that are now 1,000 times larger than small molecules and significantly more biologically complex with more structural components. And what this usually translates into is a more complex sample preparation. And in addition, there's also maybe not a competitive, but it's certainly a complementary analysis that's currently available in ligand binding assays, such as an ELISA. And there are certainly advantages to analyzing these molecules through either or both of these mechanisms. Ligand binding assays tend to have the advantage in simplicity. For anyone who's done an ELISA, it's fairly straightforward, just adding and removing liquids from a plate. And this can translate into an increased throughput capability, so where you can process hundreds to even thousands of samples in a day. In addition, ligand binding assays typically measure the free drug that's available in a sample, so things that are still, in the case of monoclonal antibodies, things that are still available for binding ligands. However, LCMS has other advantages, such as selectivity, where if you're looking for a certain peptide of a given protein, that should not have any sort of nonspecific signal that you can get with ligand binding assays, where there would be nonspecific binding. In addition, there's more flexibility in an LCMS approach, where you can look at multiple peptides of a single protein or even multiple proteins in a single sample. There's another advantage in that LCMS can be label-free. There's no enzyme-linked secondary antibody that's required to generate a signal, as there is in a ligand binding assay. There's also the advantage of reproducibility, when you can include stable isotopically labeled internal standards that can help reduce the variability that can be introduced during sample preparation. There's also an advantage in the linear dynamic range, which tends to be larger for LCMS applications, so there's less dilution of samples that is often required for ligand binding assays. So again, both of these approaches are highly valid for interrogating biologics, but it would be nice if there was a single approach that could capture the advantages of both of these. And for this, SCIEX has created its Biobe solution, and Biobe stands for Biologics Bioanalysis. Again, this was designed to essentially get the best of both worlds, where you can, the procedure uses capture antibodies that are bound to beads to essentially pull out and enrich for your protein of interest. In this case, we're demonstrating this with an antibody colored in blue. One of the advantages, obviously, is by removing the complex matrix from plasma samples. This obviously gives you a ability to see lower levels of antibody that are present in samples, and thereby increasing your LLOQ. And in addition, you can get flexibility by changing the capture antibodies that you use to interrogate your biologics. And shown here are just 3 different examples where if you use a capture antibody targeting the FC region of a monoclonal antibody, this could be used, for example, to pull out all human IgGs from an animal model, for example. If you had a drug conjugated antibody, you could actually use a capture antibody targeting the drug, which would isolate it from other human IgGs. And if you wanted to specifically measure the free antibody that was available, you could use a capture antibody targeting the complementarity determining region, or the CDR. And again, this would likely be obscured if the antibody was already bound to its target. In addition to all this flexibility, you also have the selectivity that comes from the LCMS analysis, because once they're captured, these antibodies will be digested and then analyzed by LCMS. So again, the best of both worlds. However, the workflow is often very different than what was previously experienced with small molecules, where one might be used to just a dilute and shoot approach or a protein crash approach. And so the Biobe is a complete solution that helps guide people through this fairly new and complex workflow. And again, it's to help lower the learning curve that SCIEX has sought to basically provide protocols, kit reagents, an automated sample prep solution, as well as optimized LCMS protocols. So we'll talk more about each of these as we progress. A little more information on the workflow. So basically, it's very comparable to what I've already described, where samples are combined with an internal standard, and as I mentioned, that will help with reproducibility. And then these samples and standards are incubated with conjugated beads. And the beads are included in the kit reagents. However, since this is essentially a universal kit, obviously what capture antibody a person chooses to use, as well as the corresponding internal standard, will vary by user. Once the conjugated beads have been incubated, they are brought down using a magnet because the beads are high-capacity streptavidin-coated magnetic beads. So again, high capacity indicating significant amounts of capture antibody can be included on them. And by using the magnet to pull the beads out of solution, you then wash the beads 3 times to remove the unbound proteins, and the bound proteins are then eluted using an acid. The proteins are then taken into the digest portion of the workflow, where they are reduced, alkylated, digested, and diluted. And the unique reagents for this part of the workflow is the efficient and LCMS compatible protease max and trypsin lyse C reagents. So essentially, the protease max degrades during the digestion portion, and the surfactant is now LCMS compatible, so there's no need to clean up the samples. It can actually be taken directly for LCMS analysis, so there's no desalting or dry down and resuspension. So this is a very excellent and reproducible workflow. However, as you can see from all the steps involved, it may not necessarily be, it is a fairly labor-intensive process. And so to remove some of that labor, we've actually automated the solution on the Beckman-Coulter Biomec FXP workstation shown here. The main difference is that the samples are now processed in a 96-well plate format rather than individual tubes, but the rest of the workflow remains the same. The beads are still extracted from the plasma samples using an on-deck magnet, and the proteins that are eluted are also still digested using a heating element on the deck. So everything does occur right on the deck of the instrument. In addition, since the reagents have been made into a kit, it's very easy to optimize the liquid transfers so that each experiment should be highly reproducible from run to run, and you don't get any of the variability that's introduced when different scientists prepare samples in always a slightly different way. In addition, we certainly don't expect people who have spent a long time becoming LCMS experts to suddenly become an automation expert, so we've generated a very user-friendly software that can guide you through the setup process. Really, all this entails is clicking on an icon for either the capture or the digest portion of the workflow. The software then guides you through preparing the labware that's required for the run. So essentially on the left you can see where it instructs you what volumes to put of what reagents into what positions on a reservoir, and then where that reservoir is located on the deck of the instrument shown on the right, as well as where to place tips and plates, etc. Once the system is running, it will give you updates on where in the process it is. So basically, if you do come across it during an incubation, you don't have to worry that it's not doing anything. And you can also monitor the instrument remotely. So again, you'll be ready when your samples are finished. So obviously, one of the advantages of automation is the reproducibility that can be achieved from within a run and between runs. And so the work here that will be shown was all generated on the Biomec FXP platform, and this was done by Dr. Ian Moore at SCIEX. And what he did to test the initial reproducibility was to use Rituximab spiked rat plasma, where he added 3250 nanograms per mil, which was the QC midpoint. He also added the Silamab internal standard, and this is the stable isotopically labeled universal monoclonal antibody that's sold by Sigma. And he then dispensed 50 microliters of this single sample into 93 wells of a 96-well plate, leaving 3 wells open for double blanks and a digestion control. Then pressed go on the system, and it ran through the capture and digestion protocols with a 3-hour digestion. The samples were then taken directly from the plate into the autosampler and then analyzed using the Exion LC and the QTrap 6500. And what you can see on the graph is that looking at the signature peptide across the 93 samples, you see a very nice clustering along the mean value with an intraplate CV of 8.6%. So again, a very tight, reproducible system. Particularly when considering the complexity of the workflow as well as the inherent variability in any analytical equipment. So a very reproducible system using these controls. But Dr. Moore also wanted to take this into more of a real-world scenario, and so for the next test, he used a Rituximab pharmacokinetic study, where male Sprague Dawley rats were subcutaneously dosed with Rituximab at 10 milligrams per kilogram, and blood samples were taken prior to dosing and at various time points up to 28 days post-dose. This mouse testing was done at QPS, and they also actually did an ELISA analysis of the rat samples, performing a 5 to 10-fold dilution so that they could achieve concentrations that fell within the 100 to 10,000 nanogram per mil linear range. The samples were then sent to SCIEX in Toronto, where Dr. Moore performed the Biobe analysis on those samples using 25 microliters. And he used a biotinylated anti-FC antibody for capturing the IgGs out of the rat samples, and an anti-FC antibody was also used for the ELISA, so these should be very comparable results. And what you can see is that thanks to the linear range of 100 to 100,000 nanograms per mil, there was no need to do an initial dilution of the samples. Looking specifically at the QC data, basically the lower left portion compares the double blank to the LLOQ sample, and you can see a very nice peak at the 29-minute mark, and this corresponds to roughly a 95-fold signal-to-noise ratio. So again, even at the lowest levels of quantitation, a very excellent signal-to-noise ratio. Shown on the right-hand side is the standard curve, which has excellent linearity of R value greater than 0.998. And the CVs and accuracy of the replicate values for the QCs land between 0.6 and 12.6% CVs, and accuracy between 92.4 and 108%. So a very robust and reproducible standard curve. Looking more specifically at the actual data, shown here is the Rituximab PK profile. And each point on the graph is an average of 4 rats. The red line is the ELISA averages, and the blue line are the averages as measured using the Biobe system in Toronto. And what you can see is a very excellent correlation between the two systems, particularly when considering this is 2 very different assays performed by 2 different analysts in 2 different locations over the course of a month. And to have those 2 systems be within 15% of one another is quite exceptional, suggesting that Biobe can certainly replace or certainly complement the sort of gold standard ligand binding assays. What's also of note is that the Biobe samples did come in a little lower than the ELISA, and generally this has been seen consistently. This could just be a reflection of how the capture antibodies behave differently in different assays, or it could be a measure of the increased selectivity that one gets with an LCMS approach, and that the difference may be due to nonspecific binding in the ligand binding assay. Shown here is basically just another example of the tight correlation between the ELISA and the Biobe approach, and this is showing each individual time point across 4 different rats, and again you see excellent collinearity between those two different analysis methods. And this is confirmed using a different assay measuring adalimumab. This was using a capture antibody that targeted the CDR region of the antibody. And these samples were actually performed manually, and you can still see a very excellent linear correlation between the 2 assays. It's difficult to see, but once again, the LCMS approach came in at slightly lower levels than the ELISA, again, perhaps pointing towards increased selectivity. So with that, I'd just like to summarize and say, again, hopefully we've discussed some of the challenges that can arise with LCMS analysis of biologics, but that there are also distinct benefits to using this approach, and how the Biobe solution developed by SCIEX really helps lower that learning curve and also increases the reproducibility of a given application. And that's done by, again, using consistent kit reagents, by using automation to improve the reproducibility of the sample preparation, and using the optimized LCMS protocols such as the I-methods that are available from SCIEX. And then we've discussed a real-world application looking at a Rituximab PK profile and showed how highly it was correlated with the kind of gold standard ligand binding assay. With that, I'd like to thank the people responsible, in particular Dr. Ian Moore at SCIEX, who performed all the experiments shown today, as well as the group at QPS for doing the corresponding ELISAs. And Dr. Moore has also generously agreed to join us for the Q&A session, so if there are more LCMS-specific questions, he can certainly also shed some light on those additional details. And finally, for more information, there is a link to the Biobe portion of the SCIEX website, and also a link to more general automated sample prep for LCMS that is linked to the Beckman Coulter website, and these are also available at the resource page. So with that, thank you very much for your attention.